Welcome, dear listener, to The Deep Dive, where we plunge into complex topics, break them down, and give you the essential insights you need to be truly well-informed. Today, we're embarking on a fascinating deep dive into a core concept in biology. It's not just crucial for life itself, but also, really important for your class 11th and 12th board exams. And of course, the ever-important NEET and various state-level medical entrance exams. We have a very special session today, almost like a private tuition, focusing on excretory products and their elimination.
Namaste. Absolutely. We've got a comprehensive stack of sources here to guide us, and our mission today is to extract every key nugget, clarify all the intricate details, and ensure you understand not just the what, but also the why and how of excretion. Think of this as your personalized shortcut to mastering this vital chapter, making sure every concept is crystal clear.
Okay, let's unpack this. When we think about life, we often focus on what organisms take in, food, water, air. But the constant, often overlooked challenge of life isn't just taking in nutrients, but what to do with the leftovers. How does a living being manage its metabolic waste without, poisoning itself? Today, we unravel nature's ingenious solutions, from the simplest worm to the complex human. To kick us off, we have our dedicated learner with a fundamental question.
Hello. Today I'm keen to understand the chapter on excretory products and their elimination. It seems like such a fundamental topic, not just for academics, but for understanding how living things actually survive. Where should we begin? What even are excretory products, and why do organisms need to get rid of them?
That's an excellent starting point, and it cuts right to the core of why this system exists. You see, every living organism is a hive of metabolic activity, countless chemical reactions constantly building, breaking down, transforming substances. These processes, while essential for life, inevitably produce waste products. Think of it like a factory. You produce your main product, but there are always byproducts or residues. Additionally, sometimes we ingest more than the body needs, like excess salts or water. These accumulated substances, if not removed, can quickly become toxic or disrupt the delicate internal balance of the body, a state we call homeostasis. The primary culprits we're concerned with here include nitrogenous waste like ammonia, urea, and uric acid, but also carbon dioxide, excess water, and various ions like sodium Na+, potassium K+, chloride Cl, phosphate, and sulfate. So excretion is essentially the body's sophisticated waste management system, constantly working to maintain that crucial internal stability. It's like a continuous cleaning drive, to keep our internal environment pristine.
That makes perfect sense. So it's not just about getting rid of toxins, but about maintaining balance. You mentioned nitrogenous waste, ammonia, urea, and uric acid. Are these all equally problematic, or do they differ in how difficult they are for an organism to handle?
That's a crucial distinction because, yes, they differ significantly, mainly in their toxicity and consequently in the amount of water required for their elimination. Let's take this step by step. Okay, first, ammonia. This is by far the most toxic of the three. Because of its high toxicity, it demands a large volume of water for its safe removal. Think of it like a very potent poison that needs, a lot of dilution to become harmless. On the other end of the spectrum, we have uric acid. This is the least toxic. This biological marvel allows organisms to excrete it with minimal water loss, making it highly efficient for water conservation. And then urea sits somewhere in the middle. It exhibits intermediate toxicity and thus requires an intermediate amount of water for its excretion.
So it sounds like the type of waste an animal produces is intimately tied to its environment, right?
Well,
especially how much water it has access to. Can you walk us through the different isms involved, ammonotelism, ureotelism, and uricotelism, and the specific organisms that exhibit them?
Precisely. The environment has been a powerful evolutionary driver, shaping how different life forms manage their waste and conserve water. Let's delve into these isms. First, we have ammonotelism. This refers to the process of excreting ammonia. As we just discussed, ammonia is incredibly toxic and requires abundant water for elimination. This is why you primarily find ammonotelic organisms in aquatic environments where water is, readily available. Classic examples include many bony fishes, aquatic amphibians like tadpoles, and aquatic insects. The beauty of ammonia, from an excretory perspective in water, is its high solubility. It can generally be excreted by simple diffusion directly across the body surfaces. In fish, this happens very efficiently through their gill surfaces as ammonium ions. What's fascinating here, and a point often tested in competitive exams, is that the kidneys play a very insignificant role in ammonia removal for these animals. It's mostly about this passive diffusion. Imagine a leaky bucket in a large pond. The ammonia just diffuses out effortlessly because there's plenty of water to carry away without harming the organism.
Okay, so water dwellers often just let it diffuse out. Makes sense. What about land animals?
Right. That leads us to ureotelism. This adaptation became absolutely necessary as life ventured onto land. Terrestrial animals faced a severe challenge, conserving water. Excreting highly toxic ammonia would mean losing massive amounts of precious water. To overcome this, these animals evolved a sophisticated mechanism. They convert the ammonia, which is constantly produced by their metabolism, into a far less toxic form, urea. This crucial conversion primarily takes place in the liver. Once formed, this urea is then released into the blood, efficiently filtered by the kidneys, and finally excreted. Mammals, many terrestrial amphibians, and even marine fishes are prime examples of uretelic organisms. Now, here's a detail often used to trick students in NEET. Some urea can actually be retained in the kidney matrix of certain animals. This isn't a malfunction. It's a deliberate, ingenious physiological mechanism to maintain a desired osmolarity within the kidney. This is absolutely crucial for the ability to produce concentrated urine later on. It's an example of the body fine-tuning its internal environment for optimal function.
Retaining waste to help concentrate urine. Interesting. And the third type, for really dry conditions.
Exactly. Finally, we come to uricotelism. This represents the pinnacle of water-saving excretion methods, typically found in very arid or water-scarce environments. Organisms like reptiles, birds, land snails, and insects excrete their nitrogenous waste predominantly as uric acid. Uric acid is the least toxic of the three and can be eliminated with truly minimal water loss, often appearing as a semi-solid pellet or a thick paste. If you've ever observed bird droppings, you'll notice they're not liquid, but rather a pasty white substance that's a perfect real-world illustration of highly efficient water conservation through uricotelism. This adaptation allows these animals to thrive even in extremely dry habitats, showcasing evolution's mastery of resource management.
That makes perfect sense. The environment heavily influences the type of waste, almost like nature's way of saying, here's your challenge, now adapt your waste management. I'm not sure. So is the diversity in excretory products mirrored by an equally remarkable diversity in the actual structures or organs that animals use across the animal kingdom? It seems like different creatures must have very different systems for getting rid of these things.
That's a truly insightful question, and the answer is a resounding yes. Just as organisms adapt their waste products based on their environment and metabolic needs, they've also evolved an astonishing array of structures to handle that waste. The animal kingdom showcases a remarkable evolutionary journey of excretory structures, starting from incredibly simple, almost primordial forms in invertebrates, all the way to the highly complex specialized organs we see in vertebrates, like our own kidneys. Let's take this journey step by step, starting with the simpler forms found in invertebrates. One of the classic examples you'll encounter, particularly important for your exams, are protonephridia, also famously known as flame cells. These are the primitive excretory structures found in organisms like platyhelminthes, that's flatworms, with planaria being a common example you'll study. You'll also find them in rotifers, some annelids, and even in a unique cephalocordate called Amphioxus. Their primary function isn't just general waste removal, but they are critically concerned with ionic and fluid volume regulation, a process we call osmoregulation. For those visualizing, imagine a flickering flame inside these cells. That's what gave them their name. This flickering or ciliary motion helps push fluid through their intricate system, filtering waste and regulating water.
Flame cells. Interesting name. Okay, what comes next in complexity?
Moving up the complexity ladder slightly, we find nephridia. These are the tubular excretory structures characteristic of earthworms and other annelids. Unlike protonephridia, nephridia are more advanced, directly helping to remove nitrogenous wastes and maintaining a more comprehensive fluid and ionic balance within the organism. Then, a structure that's very important for practical biology and competitive exams, especially when studying insects, are the Malpighian tubules. These are the primary excretory structures for most insects, including our common cockroach. Their dual role is crucial for survival. They efficiently remove nitrogenous waste and perform vital osmoregulation, allowing insects to thrive in diverse environments, often with minimal water.
Yeah.
And finally, for crustaceans like prawns, we see specialized structures known as antennal glands or green glands, which diligently perform their specific excretory function.
Wow, quite a variety. From flame cells to green glands.
Indeed. So you see the diversity is immense. And then when we transition to the vertebrates, this incredible evolutionary journey culminates in the development of those specialized and highly complex tubular organs, the ones central to our discussion today. The kidneys. These are truly engineering marvels of the biological world.
That's a fascinating progression. So now that we've seen the range of strategies, let's zoom in on the human system. How is our excretory system structured, and what are its main components? I recall terms like kidney, ureter, bladder, but can you give us the full breakdown, especially in an exam-oriented way?
Absolutely. The human excretory system is a beautifully designed, highly efficient filtering plant, meticulously engineered to maintain the purity of our internal environment. Understanding its structure and function is not just essential for your exams, but for appreciating the incredible precision of your own body. Let's start with the main components. It's quite straightforward. We have a pair of kidneys, one on each side, acting as the primary filtration organs. From each kidney, a tube called a ureter descends. So we have one pair of ureters. These tubes carry urine to a single muscular sac, the urinary bladder, where urine is temporarily stored. And finally, urine exits the body through a single tube called the urethra. So kidneys, ureters, bladder, urethra. Simple enough. Okay, the basic parts. Now the kidneys themselves, they're the key players, right? Tell us more about them. Exactly. Let's focus on the stars of the show, the kidneys. Let's talk about their location and gross anatomy. They are distinctive reddish-brown, bean-shaped structures, aptly described as looking like a large kidney bean. In terms of location, they are situated quite precisely in your body, positioned between the levels of the last thoracic vertebra, that's the lowest rib-bearing vertebra, and the third lumbar vertebra in your lower back. They're nestled close to the dorsal inner wall of the abdominal cavity, providing them with some protection. For an adult human, each kidney is quite a specific size, typically measuring 10 to 12 centimeters in length, 5 to 7 centimeters in width, and 2 to 3 centimeters in thickness. Their average weight ranges from 120 to 170 grams. These specific dimensions and weight, they are frequently asked in multiple choice questions in competitive exams, so make a mental note of them.
10, 12 centimeters long, 120, 170 brain chem. Got it. What about the internal structure, the hilum?
Yes, good point. Looking closer at their key features, on the inner concave surface of each kidney, there's a prominent notch or indentation called the hilum. This is a critical gateway. It's the point where the ureter, the renal artery bringing blood in, the renal vein carrying blood out, and nerves all enter or exit the kidney. Just inside this hilum, you'll find a broad funnel-shaped space known as the renal pelvis. This renal pelvis has cup-like projections extending from it, which are called calyci. A single one is a calyx. These calyces collect urine. The entire kidney is enveloped and protected by a tough, fibrous outer layer called the capsule. Internally, if you were to cut a kidney open, you'd clearly see it divide into two distinct zones. There's an outer cortex, the lighter, granular-looking region, and an inner medulla, which is darker and appears striated. The medulla isn't just a uniform mass. It's beautifully organized into a few conical structures, aptly named medullary pyramids, which project into those calyces we just mentioned. And importantly, extending inwards from the cortex, in between these medullary pyramids, are columns of cortical tissue. These are called the columns of Bertini, or simply renal columns. They are crucial for understanding the kidney's internal architecture and how its functional units are precisely organized. This intricate gross anatomy sets the stage for the microscopic marvels within.
The kidney itself sounds quite complex, with all those layers and structures working together. Cortex, medulla, pyramids, columns. But I know the true functional units are even smaller. The microscopic workhorses, right? The nephrons. That's where the real action of filtration happens, isn't it?
You're absolutely right. While the kidney provides the overall framework and macroscopic organization, the real heavy lifting, the continuous microscopic filtration and purification, is done by the nephrons. Each kidney, astonishingly, contains nearly 1 million incredibly complex tubular structures called nephrons. These are, unequivocally, the functional units of the kidney. Understanding the nephron and its intricate parts is not just central, but paramount to mastering this chapter for any exam, be it your board exams or NEET. Let's break down the nephron into its two main functional parts. We'll take this step by step for clarity. First, we have the glomerulus. This isn't just any capillary bed, it's a highly specialized tuft of capillaries. It's formed by the afferent arteriole, which is a fine branch that comes directly off the renal artery, bringing oxygenated blood to the glomerulus for filtration. Once the blood is filtered, it's then carried away from the glomerulus by a slightly narrower vessel called the efferent arteriole. This difference in diameter is key, actually, to creating the pressure needed for filtration.
Okay, glomerulus, the filter part. What's the other main part? The tubule.
Exactly. The second major part, and where the filtrate goes next, is the renal tubule. This long, winding tube begins with a unique cup-like structure called Roman's capsule. It's a double-walled, cup-shaped structure that completely encloses the glomerulus. Now, here's a very common term for exams. The glomerulus, along with its enclosing Bowman's capsule, is collectively known as the Malpighian body or renal corpuscle. Memorize that term. It's frequently asked. From the Bowman's capsule, the tubule continues to form the first major coiled section, the proximal convoluted tubule, or PCT. Proximal, because it's closest to the Bowman's capsule, and convoluted because it's highly twisted and coiled. This coiling greatly increases its surface area, which we'll see is crucial for its function. Following the PCT, the tubule straightens out and then forms a crucial hairpin-shaped structure known as Henle's loop. This loop is absolutely vital for the kidney's ability to concentrate urine. It has two distinct parts, a descending limb, which dips down into the medulla, and an ascending limb, which turns back up towards the cortex. The ascending limb of Henle's loop then leads into another highly coiled tubular region, which we call the distal convoluted tubule, or DCT. Distal because it's further away from the Bowman's capsule. Finally, the DCTs from many different nephrons, yes, multiple nephrons, all open into a single straight tube known as the collecting duct. These collecting ducts then converge even further, with many of them eventually opening into the renal pelvis, passing through those medullary pyramids and into the calyces we discussed earlier.
Wow, that's a long journey for the filtrate. Glomerulus, Bowman's capsule, PCT, loop of Henle, DCT, collecting duct. Where do these parts actually sit in the kidney zones?
Good question for orientation. The Malpighian corpuscle, that's the glomerulus and Bowman's capsule, the PCT and the DCT are all primarily situated in the cortical region, the outer part of the kidney. However, the critical Henle's loop dips down deep into the medulla, the inner region. This specific spatial arrangement, with the loop extending into the medulla, is absolutely key to its function in concentrating urine. Based on the length of Henle's loop and its dip into the medulla, we actually classify nephrons into two types, a common distinguishing point for exams. The cortical nephrons are the majority, making up about 85% of all nephrons. In these, the loop of Henle is relatively short and extends only very little into the medulla. Then we have the juxtamedullary nephrons. These are fewer in number, but incredibly significant for the kidney's function. Their loop of Henle is very long and runs deep into the medulla. This characteristic is precisely why they are crucial for the kidney's remarkable ability to produce highly concentrated urine, a process we'll explore in detail.
Cortical versus juxtamedullary, based on the loop length.
Okay.
And the blood vessels associated with them.
Right. We must mention the blood vessels intimately associated with the nephron. The efferent arteriole, carrying blood away from the glomerulus, doesn't just return to the general circulation. Instead, it branches out to form a fine capillary network that surrounds the entire renal tubule. This network is called the peritubular capillaries. A minute U-shaped vessel within this peritubular network, called the vasa recta, runs remarkably parallel to Henle's loop, especially in the juxtamedullary nephrons. This parallel arrangement is profoundly important for the countercurrent mechanism. A critical point to remember for exams is that the vasa recta is either absent or highly reduced in cortical nephrons. This serves as a classic distinguishing feature between the two nephron types and highlights their different roles in urine concentration.
Wow, that's an incredible amount of intricate parts, all interconnected. I can see why understanding each component is vital. So how do all these structures from the glomerulus to the collecting duct actually work together to form urine from the blood? That seems like the central, almost magical process of the entire system.
Excellent point, because this is where the function truly comes alive. Urine formation is a complex yet beautifully elegant three-step process. These steps occur sequentially and in different parts of the nephron, ensuring precise filtration, reabsorption, and fine-tuning of body fluids. Let's break down these three steps. The first step, the initial filtering process, is glomerular filtration. It's also often referred to as ultrafiltration. This crucial step is carried out by the glomerulus. It's the very first stage of urine production where the blood is literally screened. To give you an idea of the sheer volume involved, on average, a massive 1,100 to 1,200 milliliters of blood are filtered by your kidneys every minute.
Every minute, 1,200 milliliters, that's huge.
It is. To put that into perspective, this constitutes roughly 1/5 of the total blood pumped out by each ventricle of your heart per minute. That's a phenomenal amount of blood being processed continuously. So how does this incredibly fine filtration happen? The driving force is the glomerular capillary blood pressure. This pressure is so high that it forces blood through three incredibly fine layers, which together act as the filtration barrier. Let's visualize these three layers. First, you have the endothelium of the glomerular blood vessels themselves, the inner lining of the capillaries. Second, there's the epithelium of Bowman's capsule, the outer layer, which directly surrounds the glomerulus. And third, critically, there's a thin, delicate basement membrane sandwiched precisely between these two layers. Now, a special detail here for your exams. The epithelial cells of Bowman's capsule are called podocytes. These cells are not simply flat. They're arranged in an intricate manner, leaving minute spaces or gaps between them. These spaces are known as filtration slits or slit pores. It's through these microscopic filtration slits, through all three layers, that the blood is filtered so finely. That almost all the constituents of the plasma, with the crucial exception of proteins, are pushed into the lumen of the Bowman's capsule. This is why it's aptly called ultrafiltration. It's an extremely fine, almost molecular level of filtration.
Like a super fine sieve, leaving proteins and cells behind.
Exactly. Imagine a high-tech coffee filter. Only the liquid part, the plasma without the larger protein molecules or blood cells, passes through. This ensures vital components remain in your blood. A key metric that quantifies this process is the glomerular filtration rate, or GFR. This is defined as the amount of filtrate formed by the kidneys per minute. In a healthy individual, the GFR is approximately 125 milliliters per minute.
125 milliliters per minute, that adds up.
It does. Now, if you do the math, that translates to a staggering 180 liters per day.
180 liters, that's unbelievable. We obviously don't pee out 180 liters.
Precisely. Think about that 180 liters of fluid filtered every single day. That truly underscores the incredible efficiency and perhaps fragility of this system. Given such a high filtration rate, it's vital that the kidneys have built in precise mechanisms for regulating GFR to ensure it stays within a healthy range. One such highly efficient mechanism is carried out by the juxtaglomerular apparatus, or JGA. This is a special, sensitive region formed by cellular modifications in the distal convoluted tubule and the afferent arteriole, where they come into crucial contact. If there's a fall in GFR, meaning your kidneys aren't filtering enough, this immediately activates the JG cells to release a powerful hormone called renin. Renin then works to stimulate glomerular blood flow, thereby bringing the GFR back to its normal optimal level. This regulation through the JGA is a very common exam question for NEET, so pay close attention to its role.
Okay, JGA and renin help fix low GFR. But what about that 180 liters? Where does most of it go?
That brings us to the second major step. Remember that 180 liters of filtrate formed daily. But we only excrete about 1 to 1.5 liters of urine per day. This dramatic difference implies that a staggering nearly 99% of that initial filtrate must be reabsorbed by the renal tubules back into your bloodstream. This critical process is called reabsorption. The tubular epithelial cells lining the different segments of the nephron perform this reabsorption using either active or passive mechanisms. For instance, essential nutrients like glucose, amino acids, and sodium ions are actively transported back into the blood. This requires cellular energy, ATP, to move them against their concentration gradient. On the other hand, substances like nitrogenous wastes that we don't need and a significant amount of water are often reabsorbed passively, especially in the initial segments, simply following concentration gradients.
So reabsorption gets back almost everything useful, plus most of the water. What's the third step?
The third and final step is secretion. During urine formation, the tubular cells don't just passively reabsorb, they also actively secrete certain unwanted substances into the filtrate from the blood. These include excess hydrogen ions, H+, potassium ions, K+, and ammonia, NH3. Tubular secretion is a hugely important step because it plays a crucial role in maintaining the precise ionic and acid-base balance of our body fluids. It's the kidney's way of fine-tuning the blood's composition, ensuring that the pH and electrolyte levels remain in a very narrow, healthy range. This is critical for enzyme function and overall cellular health.
So filtration gets everything small out, then reabsorption brings back all the good stuff the body needs, and finally, secretion puts some more bad stuff back into the filtrate to be removed. That's a good overview. Can we go into more detail about what specifically happens in each part of the renal tubule? Because that seems important for the precise balancing act that leads to the final urine. Each section must have its own special job, right?
Absolutely. You've hit on a crucial point for comprehensive understanding and exam success. Each segment of the renal tubule is a marvel of specialization. Each has unique structural features and highly specific functions that contribute to the final composition of urine and, ultimately, to maintaining overall body homeostasis. This is a frequently tested area in exams, so let's delve into the where and what of reabsorption and secretion in each segment. Let's start with the proximal convoluted tubule, or PCT. This is the segment immediately following the Malpighian body, and it's quite remarkable. Its inner lining is composed of what we call a simple cuboidal brush border epithelium. The brush border refers to thousands of tiny finger-like projections called microvilli that protrude from the cell surface into the tubule lumen. This structure is no accident. It significantly increases the surface area available for reabsorption, making the PCT incredibly efficient. The PCT is truly the major reabsorption site. Nearly all essential nutrients like glucose and amino acids are completely reabsorbed here. Additionally, a substantial 70 to 80% of electrolytes like sodium and potassium and an equivalent amount of water are reabsorbed in the PCT. Beyond reabsorption, the PCT also plays a critical role in maintaining the pH and ionic balance of body fluids. It achieves this by selectively secreting hydrogen ions and ammonia into the filtrate while simultaneously absorbing bicarbonate ions, HCO3, from it. This constant adjustment ensures your blood pH stays within that vital narrow range.
PCT, the main workhorse for reabsorption and also pH balance. Okay, what about the loop of Henle?
Next, we move to Henle's loop, that distinctive hairpin-shaped structure that dips into the medulla. This loop is fundamental for the kidney's ability to concentrate urine. Let's look at its two limbs, as their permeabilities are almost opposite and a common point of confusion for students. The descending limb of the loop of Henle is highly permeable to water, but almost completely impermeable to electrolytes. As the filtrate moves down this limb, it passes into the increasingly hypertonic environment of the medullary interstitium. Because water can easily move out but solutes cannot, water passively leaves the tubule and enters the interstitial fluid, thereby significantly concentrating the filtrate as it descends.
So, descending limb water out, filtrate gets concentrated. What about going up?
Right. Conversely, the ascending limb is impermeable to water. This is crucial. However, it actively or passively transports electrolytes, particularly sodium chloride, NaCl, out of the filtrate and into the medullary fluid. As the now concentrated filtrate moves up this limb, electrolytes are removed, and since water cannot follow, the filtrate gets progressively diluted as it ascends. So the overall role of Henle's loop is absolutely crucial for setting up and maintaining the osmotic concentration gradient in the medullary interstitium. It's like a biological engine creating a salty environment, which is the key to producing concentrated urine later on. This mechanism is often tested in depth in NEET.
Descending concentrates, ascending dilutes by pumping out salt. Got it. Then comes the DCT.
Correct. Following Henle's loop, we have the distal convoluted tubule, or DCT. This segment is where the fine-tuning and precise regulation of urine composition largely occur. A key function here is the conditional reabsorption of lodium ions, Na+, and water. Conditional means this reabsorption is not constant. It is regulated by specific hormones depending on the body's immediate hydration status and electrolyte needs. The DCT is also capable of reabsorbing more bicarbonate ions and selectively secreting hydrogen and potassium ions, as well as ammonia, continuing to maintain blood pH and the crucial sodium-potassium balance in the blood. It's the last chance, really, for the body to reclaim vital substances before they're lost in urine.
DCT, fine-tuning, hormone-controlled reabsorption, more pH balance. What about the final collecting duct?
Finally, we come to the collecting duct. This is a long, straight tube that extends from the kidney cortex deep into the inner parts of the medulla. The collecting duct is the ultimate determinant of the final volume and concentration of urine. Large amounts of water can be reabsorbed from this region, especially when the body needs to conserve water. This process is under strict hormonal control. Interestingly, the collecting duct also allows the passage of small amounts of urea into the medullary interstitium. This might seem counterintuitive since urea is a waste, but it's a critical step that helps to maintain the high osmolarity of the medulla, thereby enhancing the countercurrent mechanism's efficiency. Additionally, like the DCT, the collecting duct plays a role in maintaining blood pH and ionic balance through the selective secretion of hydrogen and potassium ions. So you see, each part truly has its unique, indispensable role in this elaborate, interconnected process, ensuring our internal environment remains perfectly balanced.
That clarifies the individual functions in each segment perfectly. But how exactly does the kidney achieve the remarkable feat of producing urine that is, 4 times more concentrated than the initial filtrate? Especially since so much water is reabsorbed. That sounds like a highly complex and incredibly efficient mechanism that needs a bit more unraveling.
You've hit on one of the most elegant, ingenious, and frequently asked concepts in this chapter, particularly for competitive exams like NEET, the countercurrent mechanism. This is precisely what allows mammals, including us, to conserve water so efficiently and produce concentrated urine. It's absolutely vital for survival in terrestrial environments where water isn't always abundant. It's a testament to evolutionary efficiency. Let's break down this complex mechanism step by step, visualizing it as we go. The key players in this intricate process are the Henle's loop and the vasa recta. The genius lies in their specific arrangement and the flow of fluids within them. Firstly, consider the countercurrent flow itself. In Henle's loop, the filtrate flows down the descending limb and then immediately up the ascending limb. Notice how the flow is in opposite directions. This is the countercurrent. Similarly, the blood flow through the two limbs of the vasa recta also runs in a countercurrent pattern, flowing remarkably parallel to Henle's loop.
Okay, fluids flowing in opposite directions, side by side. Why is that so important?
The crucial element here is the proximity between the Henle's loop and the vasa recta, combined with this countercurrent flow. This close, parallel arrangement is what helps to maintain an increasing osmolarity as you move deeper towards the intermedullary interstitium. What does that mean in simpler terms? It means the fluid surrounding the tubules in the medulla becomes progressively saltier and more concentrated as you go deeper into the kidney. This concentration gradient is incredibly steep. It ranges from approximately 300 millismoles per liter, mosmol 1 in the outer cortex, right up to a whopping 1,200 mosmol 1 in the innermost part of the medulla. This fourfold increase is the engine for concentrating urine.
300 to 1,200, a huge gradient. What creates it? You mentioned salt.
Right. What causes this steep gradient? It's primarily caused by two key solutes, NaCl, sodium chloride, and urea. Let's break down their individual roles in creating and maintaining this gradient. First, the role of NaCl. Sodium chloride is actively transported out of the ascending limb of Henle's loop into the interstitial fluid, so the ascending limb is pumping salt out. This NaCl then gets exchanged with the descending limb of the vasa recta, meaning the vasa recta picks it up temporarily. As blood flows up the ascending portion of the vasa recta, that NaCl is returned to the interstitium, completing a cycle that continuously recycles the salt and maintains the gradient. It's like a perpetual motion machine for salt within the medulla, constantly replenishing the salty environment.
Okay, NaCl gets pumped out and recycled by the vasa recta. What about urea?
Urea also plays a significant part, though in smaller amounts. Small amounts of urea enter the thin segment of the ascending limb of Henle's loop from the interstitium. Then, importantly, a portion of this urea is transported back to the interstitium from the collecting tubule. This recycling of urea further contributes to and maintains that high osmotic gradient in the medulla. It's a clever way to keep the medulla highly concentrated, even using a waste product. The overall impact of this intricate countercurrent mechanism is profound. By continuously maintaining such a high concentration gradient in the medullary interstitium, it creates the perfect conditions for the final concentration of urine. The presence of this interstitial gradient facilitates the easy passage of large amounts of water from the collecting tubule. As the filtrate flows through the collecting duct, it passes through this increasingly concentrated environment, and more water is drawn out by osmosis, thereby concentrating the filtrate into the final urine. Human kidneys, as a testament to the sheer efficiency of this system, can produce urine nearly 4 times more concentrated than the initial filtrate that was formed. This is vital for water conservation, especially when you're dehydrated. For your exams, understanding the precise permeability changes in the Henle's loop limbs. Remember, descending is permeable to water, ascending is impermeable to water but pumps out solutes, and the exact roles of NaCl and urea. It's absolutely essential. Try to visualize the flow and the concentration changes. It will greatly aid your comprehension and recall. This is a classic high-yield question for competitive exams.
That was a complex but absolutely crucial explanation, and visualizing it with the roles of NaCl and urea really helps. It seems like the kidney is constantly adjusting its processes to maintain this delicate balance. How is this entire complex system, including the GFR we discussed and all that water reabsorption, regulated so precisely in the body? Are hormones heavily involved in controlling all this?
You're absolutely right. Regulation is paramount. And it's a highly sophisticated interplay of hormonal feedback mechanisms, ensuring your body's vital filtration system never falters. It involves not just the brain's hypothalamus and the JGA, which we briefly touched upon, but also, surprisingly, signals from the heart itself. This entire regulatory network is another high-yield area for competitive exams, so let's delve into it in detail. First, let's talk about regulation by the hypothalamus, primarily through ADH or vasopressin. Your body has specialized sensors called osmoreceptors. These are like tiny internal monitors constantly checking for changes in blood volume, overall body fluid volume, and the concentration of ions in your blood. For example, if you're experiencing excessive fluid loss, perhaps from sweating a lot on a hot day or not drinking enough water, these receptors immediately spring into action. Once activated, these osmoreceptors send urgent signals to the hypothalamus in your brain. The hypothalamus then stimulates the neurohypophysis, the posterior pituitary gland, to release a crucial hormone called antidiuretic hormone, ADH, also known as vasopressin.
ADH, antidiuretic hormone, so it stops you peeing too much.
Essentially, yes. The effects of ADH are central to water conservation. Here's what it does. It primarily facilitates water reabsorption from the latter parts of the tubule, specifically the distal convoluted tubule, DCT, and the collecting duct. It makes these parts more permeable to water, allowing more water to be drawn out. By increasing water reabsorption, it actively prevents diuresis, excessive urine production, thereby conserving precious water for the body. ADH also has another important function. It can cause constriction of blood vessels throughout the body. This constriction leads to an increase in overall blood pressure, which in turn can increase glomerular blood flow and subsequently the GFR. This entire process works on a finely tuned negative feedback loop. If your body fluid volume increases, say, after drinking a lot of water, it switches off those osmoreceptors, which then suppresses ADH release. This brings the system back to normal and allows you to excrete excess water.
Okay, ADH controls water reabsorption based on hydration. What's the next system? The renin-angiotensin one.
Correct. Second, we have the incredibly important and complex renin-angiotensin mechanism, often called the RAAS pathway. This mechanism acts like your kidney's emergency response team, ready to spring into action. It's primarily initiated when there's a fall in glomerular blood flow, glomerular blood pressure, or the GFR itself, basically, when your kidneys aren't getting enough blood flow or filtration pressure. When this critical drop occurs, the specialized JG cells in the juxtaglomerular apparatus, JGA, become activated. These activated JG cells immediately release the enzyme renin. Renin then acts swiftly in the blood, converting a protein called angiotensinogen, produced by the liver, always circulating, into angiotensin IV. Angiotensin again isn't the final active form. It circulates to the lungs, where it's further converted by an enzyme called ACE, angiotensin-converting enzyme, into its highly potent form, angiotensin II.
Renin makes angiotensin the first, then ACE makes angiotensin second. What does angiotensin the second do?
Angiotensin II is a truly powerful molecule with multiple effects vital for regulation. Firstly, it's a very potent vasoconstrictor. It causes blood vessels to narrow. This directly increases systemic blood pressure, which in turn increases the glomerular blood pressure and consequently boosts the GFR. Secondly, angiotensin II acts on the adrenal cortex, the gland sitting atop your kidneys, activating it to release another crucial hormone called aldosterone. Aldosterone then causes increased reabsorption of sodium ions, Na+, and water from the distal parts of the renal tubule, specifically the DCP and collecting duct. This reabsorption also directly contributes to an increase in blood pressure and GFR. So this entire complex cascade renin, angiotensin I, angiotensin II, aldosterone aims to rapidly restore normal blood pressure and GFR when they drop, ensuring vital organ perfusion and filtration.
Wow, a whole cascade to raise blood pressure and GFR. Is there anything that lowers it?
Yes, absolutely. Nature loves balance. And finally, let's consider the atrial natriuretic factor, ANF mechanism, which originates directly from the heart itself. This mechanism acts as an important counter-regulatory system to the renin-angiotensin pathway, preventing blood pressure from becoming too high. When there's an increase in blood flow to the atria of the heart, for example, due to increased blood volume putting more stretch on the atrial walls, it causes the specialized atrial cells to release atrial natriuretic factor, ANF. The effect of ANF is quite opposite to angiotensin II. It causes vasodilation, the dilation of blood vessels. This vasodilation leads to a decrease in blood pressure. Therefore, the ANF mechanism essentially acts as a crucial check or brake on the renin-angiotensin mechanism, preventing an excessive or uncontrolled increase in blood pressure. These three systems, ADH, renin-angiotensin, and ANF, work together in a finely tuned dynamic balance to maintain stable kidney function and overall fluid and electrolyte homeostasis in your body. It's an incredible symphony of hormones and feedback loops.
So many hormones and feedback loops. It's truly like a finely tuned machine, constantly adjusting to keep everything balanced. What happens once the urine is actually formed and concentrated through all these processes? How do we actually, get rid of it from the body?
That brings us to the final step of waste elimination. Micturition, which is simply the scientific term for the process of urination or voiding the bladder. This isn't just a simple emptying, it's a fascinating neuromuscular reflex that allows us to voluntarily control the release of urine after it's been formed. Let's trace the path. Once urine is formed by the millions of nephrons, it flows out of the collecting ducts into the renal pelvis, then down the ureters, and is ultimately carried to the urinary bladder where it is temporarily stored. This storage continues, with the bladder gradually filling until a specific signal is given by the central nervous system, or CNS. This signal is primarily initiated by the stretching of the urinary bladder walls as it gradually fills with urine. As the bladder expands and distends, specialized stretch receptors located on its walls detect this stretching and send nerve signals back to the CNS, specifically to the spinal cord, and then up to the brain.
Stretch receptors signal the brain bladder fault. What does the brain do?
In response to these signals from the stretch receptors, the CNS processes the information and then sends out coordinated motor messages. These messages have two crucial simultaneous effects. 1. First, they initiate the contraction of the smooth muscles of the bladder wall, called the detrusor muscle. This contraction creates pressure, pushing the urine out. 2. Second, and concurrently, they cause the relaxation of the urethral sphincter, which is a muscular ring that normally keeps the urethra closed, preventing leakage. The combined effect of bladder contraction and sphincter relaxation leads to the controlled release of urine from the body. The entire neural mechanism causing this coordinated process is collectively called the micturition reflex. While it's a reflex in adults, it's largely under voluntary control, meaning we can consciously inhibit or initiate it unless the bladder becomes excessively full. It's interesting to note some characteristics of urine in a healthy adult human, as these can be important diagnostic indicators. On average, a healthy adult excretes about 1 to 1.5 liters of urine per day. The urine formed is typically a light yellow-colored watery fluid, which is slightly acidic with a pH of approximately 6.0, and it has a characteristic odor. On average, about 25 to 30 grams of urea are excreted per day. Finally, it's worth knowing that analysis of urine is actually a powerful and non-invasive tool for clinical diagnosis of many metabolic disorders and kidney malfunctions. For instance, the presence of glucose in urine, a condition known as glycosuria, is a strong indicator of diabetes mellitus. Similarly, the presence of ketone bodies in urine, termed ketonuria, also frequently suggests diabetes mellitus. These are common diagnostic markers doctors look for in a routine urine test, showcasing the practical application of understanding this system.
It's incredible how the kidneys are the main filtration system, but earlier you hinted that other organs also contribute to waste removal. Are there other organs in the body that also contribute to eliminating waste, even if it's not their primary function?
That's an excellent follow-up, and you're absolutely right. While the kidneys are undoubtedly the primary dedicated excretory organs, other organs also play significant supplementary roles in waste elimination. This is often overlooked in academic discussions, but is important for a holistic understanding of how the body maintains its internal environment, its homeostasis. Let's consider a few. First, the lungs. While their primary role is, of course, gas exchange, taking in oxygen and expelling carbon dioxide, they are also active excretory organs. They efficiently remove large amounts of carbon dioxide, approximately 200 milliliters per minute. In addition to CO2, the lungs also eliminate significant quantities of water every day in the form of water vapor when we exhale. So every breath you take is also a subtle act of excretion.
Lungs get rid of CO2 and water vapor. What else? The liver.
Yes. Next up is the liver, which is actually the largest gland in our body and a metabolic powerhouse. The liver plays a massive role in detoxification, processing, and breaking down various substances. It secretes bile, and this bile contains various waste products specifically meant for elimination. These include substances like bilirubin and biliverdin, which are the breakdown products of hemoglobin from old red blood cells. Bile also contains cholesterol, degraded steroid hormones like estrogen and testosterone after they've served their purpose, certain vitamins, and even drugs or medications that a body needs to get rid of. Most of these substances, once secreted into the bile, ultimately pass out of the body along with digestive wastes in the feces. So the liver's role in chemically processing and packaging these wastes for removal is absolutely crucial.
Liver packages waste into bile for removal via digestion. What about the skin?
Our skin also contributes through two types of glands that you might not immediately think of as excretory: sweat glands and sebaceous glands. Sweat glands produce sweat, which is a watery fluid. While the primary function of sweat is cooling through evaporation, it also serves an excretory role. Sweat contains not only water but also sodium chloride, NeCl, small amounts of urea, lactic acid, and other substances. So when you sweat, you're also helping to excrete some metabolic wastes, albeit in smaller quantities compared to the kidneys. Then there are the sebaceous glands. These glands eliminate substances like sterols, hydrocarbons, and waxes through their oily secretion called sebum. While sebum's primary function is to provide a protective oily covering for the skin, keeping it supple and waterproof, the substances eliminated in sebum are essentially waste products of metabolism.
Sweat and sebum also remove some waste. Any others?
And finally, a lesser-known fact, but quite interesting, is that small amounts of nitrogenous wastes can even be eliminated through saliva. While this is a very minor route compared to the kidneys or even sweat, it demonstrates just how many different pathways the body uses to maintain its internal purity and balance. It's a true testament to the body's comprehensive waste management strategy.
It's truly incredible how many organs contribute to this vital process, all working together. But what happens if the kidneys, which are clearly the main filtration system, fail or malfunction? What are some common disorders that can arise if this finely tuned system goes awry? And what can be done about them?
That's a critical aspect to discuss, especially for understanding the clinical implications and the profound impact of these systems on overall health. Kidney malfunction can indeed lead to severe health issues, and unfortunately, several common disorders can affect them. Understanding these is important for a holistic view and often comes up in your exams. One of the most serious conditions is uremia. This occurs when there's an accumulation of urea, that primary nitrogenous waste, and other nitrogenous wastes in the blood. Given that urea is toxic, its buildup can be highly harmful and can rapidly lead to kidney failure, if not addressed promptly. For patients suffering from severe uremia, a life-saving treatment is hemodialysis, often referred to as using an artificial kidney.
Hemodialysis. How does that actually work?
Right, let's understand hemodialysis in a bit more detail, as it's a marvel of medical technology. The process involves drawing blood from a convenient artery. Before this blood enters the dialyzing unit, an anticoagulant like heparin is added to prevent clotting. Dialyzing unit. This blood is then pumped into the dialyzing unit, the artificial kidney. Inside, there's a long, coiled, semi-permeable tube, typically made of cellophane, surrounded by a special fluid, the dialyzing fluid composition. The key is its precise composition. It has the same composition as normal blood plasma, except for the nitrogenous wastes. This difference is crucial. Mechanism. The porous cellophane membrane allows molecules to pass based on concentration gradient. Since nitrogenous wastes are high in the patient's blood but absent in the dialyzing fluid, these substances freely diffuse out of the blood, clearing it. Return. Once the blood is cleaned, antiheparin is added, and the cleared blood is pumped back into the patient's body through a vein. This method has truly been a boon for thousands of uremic patients worldwide.
So dialysis filters the blood externally. What if the kidneys fail completely?
While hemodialysis is life-saving, the ultimate and most definitive method for correcting acute or chronic renal failures where kidneys permanently lose function is kidney transplantation. This involves surgically implanting a functioning kidney from a donor into the patient's body. To maximize success and minimize rejection risk, the donor is preferably a close relative, as their tissues are genetically more similar. Thanks to immense advancements in modern clinical procedures and immunosuppressive drugs, the success rate of this complex technique has significantly increased, offering new hope.
Transplantation is the ultimate fix. Are there other common kidney problems, like stones?
Yes. Besides these severe conditions, there are other common disorders. One is renal calculi, commonly known as kidney stones. These are essentially stone-like or insoluble masses of crystallized salts, often oxalates like calcium oxalate stones, phosphates or uric acid, that form within the kidney. They can cause excruciating pain and block urine flow. Another disorder is glomerulonephritis. This is a condition characterized by the inflammation of the glomeruli of the kidney. As we discussed, the glomeruli are the primary sites of blood filtration. So inflammation here directly impacts the kidney's filtration capacity, leading to impaired kidney function. So we've journeyed through the entire process today, from understanding the different types of excretory products and their elimination strategies, to taking a deep dive into the intricate human excretory system, its anatomy, the amazing nephron, the precise three-step process of urine formation, the wonders of the countercurrent mechanism, the complex hormonal regulatory mechanisms, and finally the disorders associated with kidney malfunction and their treatments. Remember, this entire system is brilliantly designed for maintaining homeostasis, the internal balance of our body. Every single detail, from the podocytes to the countercurrent, plays a vital, interconnected role in keeping you healthy. For your exams, I strongly recommend you revise this chapter again, paying special attention to the diagrams and flowcharts of urine formation and the regulatory pathways. Practice past year questions, especially those on GFR regulation and the countercurrent mechanism. They're very common in NEET and often differentiate strong candidates. And always remember, if you have any lingering doubts on this chapter or any other topic, please don't hesitate to reach out. Keep learning, keep questioning, and keep aspiring.
What a truly comprehensive and crystal clear explanation. That deep dive truly pulled back the curtain on how intricate and interconnected our internal systems are, right down to the microscopic level of the nephron. It makes you realize just how finely tuned homeostasis is, how our bodies constantly manage waste while preserving everything vital. It's astonishing how something we often take for granted, like urination, is the culmination of such complex, elegant biological engineering. And that wraps up another deep dive for you, our dedicated learner. Until Until next time, keep exploring, stay curious, and keep connecting those dots.